Good morning, everyone. My name is Richter. I'm a second-year doctoral student here at Northeastern in the Social Science Environmental Health Research Institute. And our panel right now is on social science in government, regulatory, and research agencies. We will have two speakers, and each of them will speak for 15 minutes, and then we'll do questions. Our first speaker is Cecile Wendling from CNRS-Sciences, Po, Paris. Our second speaker will be Rachel Morello-Frosch from the University of California-Berkeley.
Hi everybody, and thank you for having me here. And it has been fascinating and very inspiring for me to listen to all of you. So I just wish I could give you a lot of things back that also inspire you. I'm going to talk about a project which was a collaborative project. I've done this project with Olivier Boraz, Daniel Benamouzik, Daniel Salomon, and Jean-Noël Jouzel. This project was commissioned by the French agency in charge of environmental risk, occupational risk, and food safety. This organization is a public organization, is financed by 4 different ministries, and it's working on the basis of questions, either questions coming from the government, questions coming from communities, or questions coming from the agency itself. So a question could be, for instance, what about fire retardant? Should we put them in sofas and beds in France or not? And then how it's working, the agency asks for the organization of a working group, so they recruit academics. They give them a certain time period, so I don't know, 6 months, or it can be 3 months if it's an emergency. And then they have to come out of the work with a recommendation and a report that is assessing the risk. And based on this report, then the ministry can manage the risk, so decide what to do. So when I arrived at this ANSES, you must know that those working groups were mainly composed of what we call hard scientists. So 90 to 95% of those working groups were made out of toxicologists, epidemiologists, engineers, people doing chemistry, and so on. There were one very small department in ANSES composed of 4 social scientists and called Risk and Society. That's the name of the department. And they wanted to push for the inclusion in those working groups of social scientists. So it could be a psychologist, it could be a sociologist, it could be a lawyer, it could be somebody doing economy. And they found it really difficult. So what they have done, they have commissioned the CSO to work, and there are 2 parts of my work. First of all, being present every day in this ANSES during 9 months, watching the meetings, the expert groups, and see, do they manage to work together? When there is 1 or 2 social scientists with 12 or 15 other scientists, how do they share their knowledge and so on? But more interestingly, they also wanted to know, what do other agencies do? So in Canada, or in the US, or in Germany, in the UK, can we learn from them? And so this is how we decided to come out with a methods based on interviews, which I will present. Then I will give you, so the interviews were conducted to understand the reason why social scientists were used by those public agencies in charge of risk. Then I will explain the different choices that have been made in how to use the social science, and then give some conclusion. So we decided to focus on UK, Germany, Netherlands, US, and Canada. And I must say that it was quite difficult, first of all, to find counterparts to ANSES, because it's kind of particular to the French system to have such an agency. And second of all, it's really difficult to find the social scientists in each of those organizations. You have an epidemiology department, or you have a department for water safety, but when you really look for the social scientists, most of the time it was not so easy. So the first set of questions I had was really, why do you use social science? And that's really interesting, because some of the people said to have a more comprehensive approach of the risk. So they said that the reason why they were hired inside the agency was that because people realized that the risk was something else than the figures or the limits or the thresholds. A second comment that always came out was understanding the uncertainties and the ignorance. And so it was really interesting for me, because sociology of ignorance is very important, to see that people inside agencies had identified that there were blind spots and that they didn't know how to deal with it. And also they were saying that that's for the method. We need more robust models, and to understand the limit of a model, being able to express better ways the assumptions behind the model was helping them. The second reason they always say is the link with society. We need to understand the people, their perception, their fear. We need to know how to influence their behavior so that they live in a safer way, and we need to include the civil society. And this was really important to them as agency, because when they don't include them, then there are reports in the press and people go and claim that this decision was not good and so on. So the question is, how do we work with them? The third reason why really science and values. We need to be ethical when we conduct our research projects, and we need to know how do we use the precautionary principle, how do we distinguish between the scientific debate and political debate. And the last thing was the organizational aspects. So do we better understand the context of a risk, and do we better understand how we are internally organized to assess the risk? So as you see, it was really broad, and people on the phone didn't have the same view on the reason why social science were called in. And then I found out two models. There are those kind of agency where they have what they call multidisciplinary approach. So most of the time, for instance, on the fire retardant is a good example, then you will have a risk assessment report on one side, when they only focus on the toxicology and the epidemiology and those kind of things, and the concern assessment report. And the concern assessment report was interview with the population, the communities, the workers producing the products, the distributors of the product, the people using the product, and so on. And this was most of the cases I found that was like this. So there were two separate reports. So I would say hard science on one side and social science on the other side. And then there are people trying to do really what they call transdisciplinary work. So really embedding the social scientists within the laboratory. And an example were cases around synthetic biology, for instance, where they really work together. Another choices that I always found was either those agencies, they internalize the social scientists. So, for instance, IRVM in the Netherlands or the BFR in Germany, they have social scientists inside the organization. They are paid by the organization. And that's their main work. And in Italy or in Canada, they do not have internal research scientists. What they do is that they do call for projects on one particular issue, and they call for their expertise. And then they just give their expertise for the project, and then they continue their university work. Then I interviewed the social scientists themselves on the roles that they think they had in their organization. And so in RIVM in the Netherlands, it was really about revealing implicit, tacit aspects of risk assessment and risk management. So I think they are kind of Cassandras, so they see something that others don't see most of the time. And they try also to nuance the results and the presentation, give more information on the process, on how it was produced. At the HSC in UK, it was more about contextualize, like we said yesterday, context, 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 so it was kind of this. And the FSA, it was interesting to me because they were here to guarantee the good use of methods and good use of data. So they were not used to give impact on the result of the project, but only on the process. You know, are the samples good? Do the people that we interview have a consent, and so on. So to me, it was really interesting because in some of the places where we're talking, there were only process guys or methods guys only. And in some others, there were only opening up guys, so people going away in different communities to see what is the context. And some others, they were really working deep inside on the bias of the research process itself. Other roles that they claimed to have was to be translator, so to enhance the communication between the stakeholders and the agency, or between the general public and the agencies. In Canada, they felt they were representative of the minorities, that they were speaking for the minorities. they claimed that before the inclusion of social science, they were conducting report for a given individual, like what we'll say a lambda individual, somebody like taken by random. And after the use of social science, they say, no, we have to use different classification of groups and individuals, and some of them have inequalities, and some of them are maybe more at risk than others. They say also they were educators, so they wanted to educate people to risk. In Austria, I interviewed people that said that social science can also help in strategic foresight. So they were working in anticipating the risk of tomorrow. So what would be the next molecules that would be a risk for the society that we do not talk about today, but that are going to come up on the agenda tomorrow? There were also in Canada what I call ex-post analyzer. So after a project was finished, sometimes only with toxicologists, epidemiologists, they will look at the process and try to see the lessons. What went wrong, what went right. And finally, some of people say, well, I have no role at all in the risk expertise process. I only do fundamental research, but I will not give a recommendation as such to the agency. So then I looked, after talking about their role, I looked at the different outputs, and what I found is most of the time it's a report on the social aspects of risk. And this report is made out of a literature review, survey, focus groups, discourse analysis, observation. And those reports are a part of a big process of assessment of a risk. Another way to do it is not to do a report, but to have kind of a mini checklist, or what they call a quick scan, of the social aspects to be taken into account. Because people in the agencies, they don't know what is sociology. They don't even know what a sociologist could do for them. They don't know what type of question they could ask. So then, if there is kind of a checklist, preliminary checklist, for instance, are there inequalities in this topic? Or are there high political interests? Are there economic interests on board? And those kind of checklists, have there been a paradigm shift in the science studying the topic? Those kind of checklists. Then they knew that the questions could be raised, those type of questions could be raised to the social scientists. So this is interesting to me to say that, because they try to see out of the box of the report what they could do. And this is how the checklist came out. The question then is how this checklist can be used, and we can talk about it later. I'm going out of time then. So then I looked also at the organizational aspects. Where are the social scientists? And that was really different from one place to another. So sometimes they are in the strategy department, so really involved in the process to conduct the science. Sometimes they are in the ethical department, just to make sure that there will not be people claiming that it was not right the way the process was done. A lot of time, for instance also in Germany, it's in the communication department. And that was, to me as a sociologist, really scary, because they didn't think that a sociologist had something to say about the risk itself. But you know, at the end, when we have done everything without you, we need to know how do we communicate about this. But it's too late, you know. Actually, you should have think of this from the beginning. So, yeah, I have 3 minutes, and I realize it's too long, but what I want to show with this presentation is that there is no institutionalization of the way people use social scientists today. There is no clear definition of the role, and it's kind of an experiment going on. So each agency tries to figure out, oh, maybe I could create a social science panel. Instead of having one social scientist in every working group, will it work better? Or maybe I could have both internal and external scientists, social scientists. Would it work better? So to me, this work was exploring those possibilities. And we had a workshop in Paris, and then 1 year later, one in London, where all the people participating in those interviews could come and gather and share their thoughts about their different roles and the different difficulties. We created on LinkedIn a Paris Risk Group. It's called Paris Risk Group because the first meeting was in Paris, where people exchange about their problems, their methodologies, the quality of their work, the resources that they could share. And the only places where really progress were made between these fieldwork and now, when the head of the organization, so the head of the agencies, had a social scientist background, or somebody doing social science like his wife or his daughter, somebody really a personal connection to social science. Else, they do not even understand why they have social scientists. The solution, the best study were found when there were people with double curriculum options. So people that were both environmental health specialists and social scientists. And to me, this is really about curriculum and the training of the people. The existence of a crisis, so the BSc crisis or any type of environmental crisis that we could have, pushed for the organization to recruit social scientists. So there is always a window of opportunity after a crisis. And only with training of non-social scientists can it work. And I will finish with two small anecdotes. The first one is a meeting at the ANSES, and a medical doctor said, "Oh, there is a sociologist in the room." What the hell? You know, how can a journalist or a sociologist think that he has something to say about risk? That's nonsense. And it was one of my first week. I was like, ooh, that's a good start, interesting. And the second meeting I had, which was also very interesting to me, we had tuberculosis, bovine tuberculosis in France. And the way the question was framed was not giving an options for an anthropologist, for instance, to go and see how people breed and how they feed the animals, not at all. It was only seen from a hard science perspective, and the conclusion was kill the badger, because the badger was a small animal contaminating the farm. And I was sitting in the room and thinking, oh my God, that's so short term, because next time we will have a question from the government because of a fly or a mosquito, and the one eating it was the badger, of course. And I could not, and then when there were social scientists, I asked them, but what can you do? And they say, well, the problem is the framing of the question. If the framing of the question right from the beginning doesn't not include like field work, going to see how it's really happening, and doesn't include experiencing the risk with the people that suffer from it and so on, the answer will be always in the black and white, like kill or not kill. So with those two anecdotes, I would like to stop, and to me, you're doing an amazing, you have an amazing role to play, not only where you are, but contributing to the work of those agencies. My experience also is that they're looking for social scientists to apply to their working group, and they do not have a lot of them. And I can understand why they do not have a lot of them, because for them it's hard. They arrive in a ground where people do not speak their language, do not ask questions in a way that they could contribute. So it's really like some effort has to come from the agencies, and some effort has to come from the social scientist community. Thank you.
Hi, everybody. I'm Rachel Morello, and I want to shift gears a little bit and talk a little bit about a project that we did as part of our NIEHS Ethics Collaborative on doing co-production of results communication protocols for a biomonitoring pilot study that was carried out through California's biomonitoring program known as Biomonitoring California. So I want to give you a little bit of background and then talk about the project, and then discuss a little bit its implications. So I just want to reiterate for this room the extent to which what I like to call toxic ignorance is really baked into our regulatory system. If you look at some of the chemicals that are manufactured or imported into the U.S., at more than 25,000 pounds per year, only about 200 have been adequately tested. And I was thinking this morning when people were talking, it is kind of ironic how we spend a lot of time, us scientists, trying to get grants to figure out and do exposure assessment and biomonitoring and all this kind of stuff to find out what's in people when industry kind of knows damn well what's in their products, they're just not telling us. So if we want to do a cost-benefit analysis of that sort of issue, anyway. So I'm in this business too, to do biomonitoring, and right now we're in a situation where the lack of data or our toxic ignorance, no data tends to mean no problem in the regulatory arena. And as a result, we have extensive exposures that occur through different routes and through different experiences in the different environments in which people live, work, and play, as environmental justice advocates like to say, and also through the products that we use in our homes and slather on our bodies every day. And so biomonitoring technology has been a strategy that has been used to try and quantify human exposures to environmental chemicals. And over the last 10 years, the analytical techniques have really improved drastically and very rapidly. So we can now detect more and more chemicals at lower and lower levels. In addition, a lot of these techniques are becoming more affordable and accessible to a diverse array of stakeholders. So it's not only the purview of academic scientists anymore, you have a lot of different groups that are using these technologies and techniques. And not surprisingly, our technological know-how has outpaced our ability to interpret what we find in people's bodies. So we're very good at showing what we found and how much, as Julie said in her presentation yesterday, but we're not necessarily good at answering the rather straightforward question of what does it mean for our health. And sometimes we have difficulty even explaining to people what the primary sources of their exposure are. And in one of our interviewees who participated in a biomonitoring study said, none of these chemicals comes with a return address. So, and then people have mentioned IRB issues, and our group has really sought to grapple with these. And so I'm sure people are familiar with the Belmont principles: respect for persons, beneficence or do no harm, justice, all of which are important to keep in mind. And I think IRB, generally academic IRBs, I think this is shifting a little bit in part because of a lot of the work being done in this room to educate IRBs, both academic and otherwise. But they still tend to apply sort of a traditional clinical ethics approach when they're reviewing environmental health research projects. So there's a lot of concern about whether and how we share individual exposure results and biomonitoring results with study participants when we can't really tell people what the individual level health implications are of what we find. So telling people that we found 46 chemicals in your blood sample and then not being able to answer the logical next question, is it high, will it make me sick, that kind of stuff. IRBs tend to assume that reporting uncertain information is likely to cause participants undue worry, stress, or harm. And so generally what IRBs tend to prefer, and what many scientists do, is they may a priori decide whether certain chemical exposure results are clinically relevant when there is some kind of benchmark. So lead is a very good example of that, where there is a very sort of clear benchmark that is applied in clinical settings. And so when levels exceed clinical action levels, then we might report back. For example, the CDC and Haynes does this for things like metals, lead, and mercury, etc. However, we have come to find that there's problems with the application of the clinical ethics model. One is that clinical information and communication in the clinical setting generally is changing a lot. There is a trend towards telling patients more and more about their health data, and there's a lot of uncertainties inherent in that. There's been some really good projects, for example, the Open Notes experiment, which is basically making doctors' notes available to participants, and there's a lot of uncertainty about how you interpret that. There's traditional notions of clinical ethics that is not really true anymore. The other thing, there's a lot of lessons in the fields of genetics and and brain imaging research that is proliferating as well. And studies of participants in those studies make really clear that they want their information even if the results are unclear. Increasingly, participants are making that as a condition of their willingness to participate in research. They want to know what kind of information are you going to give me. The other thing is clinical ethics, I think, underestimates, or we believe our group, that it underestimates people's ability to grapple with scientific uncertainty. It's a little bit patronizing, I think, that for some reason participants can't, deal with uncertainty and they're going to get stressed out about it. And probably most important is that if by not reporting, we are precluding people's ability to take action to reduce their exposures. And if you apply clinical action level history, for example, for lead, what was clinically actionable 10 years ago is much lower now. So clinical action levels evolve and get lower as our biomonitoring gets better. We find that lower and lower levels of chemicals can result in adverse health effects. So we don't want to preclude people's ability to do that. So we have proposed these notions of post-Belmont ethics in that we want to consider individual rights as well as community rights to be considered. IRB still, I think, really emphasize individual rights. We want to be able to address this tension that Julia alluded to yesterday between the people's right to know their results, but also their right that they're taking into consideration their ability to act. Okay, you can give people a lot of information, but for example, in certain occupational contexts, people do not have the freedom to change their working conditions to reduce their exposures in certain cases. So how do we acknowledge that? And that requires, really working with potential study participants and communities to help you play a role in human subjects protection and in the development of study protocols a priori as much as possible to kind of navigate those tensions that may or may not be completely resolvable, but at least there's a deliberative process to figure out how you could potentially deal with them. So I wanted to give you an example of how we worked in a collaborative with Biomonitoring California, which is, again, California's biomonitoring program. It's run by the California Department of Public Health and the Department of Toxic Substances Control. It was passed in large measure because of the hard work of activists, including Charles, the Breast Cancer Fund. A lot of environmental health activists made this happen. And so Biomonitoring California, when it was passed, was also passed the idea was to create a biomonitoring program similar to the one that CDC had, but for California. And it was passed at a time when then the Great Recession hit and we had a $20 billion deficit. So we were not going to have the infrastructure to do this. And so instead, they became entrepreneurial. The program decided to do pilot studies. And so this was one that we worked on with them called Chemicals in Our Bodies. And we recruited and tested 90 mothers and their newborn infants for exposures to environmental chemicals, and where possible, trying to identify sources of exposure through interviews. But one of the main strategies was to develop a protocol for informing participants about their biomonitoring results. And that's what I'm going to focus on today. So we recruited women as they were delivering at San Francisco General Hospital, and they were kind enough to do in-person interviews. They did take-home questionnaires. They gave us a blood sample, and they also gave us a urine sample, and where possible, we collected cord blood at delivery. And during that period between sample collection and when we were going to deliver results, we wanted to work with participants to try and develop a useful protocol to return results to participants who wanted them. California's biomonitoring law it's required in the law that anyone who participates in any of their biomonitoring projects has to have access to the results if they want them. So they are required to return results. So they did the program did not have a choice. It was not whether, but how we're going to return results. And so we did a usability testing strategy to develop this, which I'll explain to you in a little bit. But we don't have a pointer here, do we? So This is looking at some preliminary results in our study sample. This is just for blood. So we had 77 maternal blood samples and 55 cord blood samples, and these are just looking at the number of chemicals that we found out of a total of 59 in each individual participant. And so it ranged from 27 to 45 in the women with a median of 37 chemicals, and it ranged from 21 to 43 with a median of 32 chemicals in the cord blood. This is just for the blood results. And a lot of our motivation for report back again was because the law required it, but also we were asked to help them in part because of our experiences with the Cape Cod Household Exposure Study and the Northern California Household Exposure Study in Richmond, which Julia talked about yesterday, and also just a general desire of the health department, the biomonitoring program, to advance biomonitoring literacy among people who were in their studies. So the demographics of the population was that it's overwhelmingly Latinas. This is a safety net hospital for anyone who does not have health insurance. You can get excellent prenatal access and delivery services. So many of the patients are low income, and over half were Spanish speakers. And in general, people had little knowledge about environmental chemicals and environmental health. And just to reiterate the questions that participants are always interested in knowing, that Julia talked about yesterday, these participants have the same questions. We developed a packet, and the idea was to develop a protocol. And so it was decided to do this through mail. And it was going to contain a cover letter, some kind of textual summary of results, visuals, and then a list of chemicals that were tested. And it was going to be organized in chapters by chemical class. And so the idea was to develop a prototype report back packet, invite participants to sit down with us, look through this packet, and we'd say to them, we want you to pretend this is your packet. Okay, you get this in the mail, this is your data. Tell us, how does this work for you? Is this good? What do you understand? What are some off-putting things? So we really wanted to know about the messages. Is it linguistically appropriate? Is it culturally appropriate? What are some things that are missing that you would like to see here? So participants have a long time to stare at the materials, go through them. We ask them to mark it up. And then when they're ready, we do a semi-structured interview with a script to kind of systematically assess how they are understanding and interpreting the materials that they're looking at. And again, we're asking them to assume that the data that's in these materials is theirs. It's not theirs yet because the results were not ready. And we take a lot of notes during the process and record the interviews. So I just want to show you how input from study participants themselves can really have a huge impact on how you design your protocols. I also want to emphasize this was not a community-based participatory research project. These were people who were just randomly recruited as they were delivering in a hospital. But I want to emphasize that even in that context, you can really engage your study participants in the development of your protocol. There are opportunities even within that context to do this. So I'm going to show you the initial versus final summary results so you can see how feedback really changed things. Overall, people really wanted shorter sentences. They want more white space. They want information in a question and answer format. They want the navigation between the documents to be a little more clear, and they want simple tables, and they ask for us for a lot of reformatting. So this is what we started out with as our draft protocol. That's what we asked people to tell us what they thought about. And in part, this was a lot of negotiation with different government agencies who are often concerned and want to put in as much text as possible, so there's always that tension. So after people, this is looking at metals in blood, this is for lead. We're trying to tell people where lead is found, what are the possible risks, and how they could reduce their exposure with a little summary at the beginning, what kind of metals we found and how many we tested. How many people we tested. So after they looked at these materials, and we did several rounds of usability testing in both English and Spanish, this is what we ended up with. We got rid of a lot of text. We completely reformatted this, and so this is what the materials ended up looking like. Instead of being all crammed onto one page, they wanted this first page, and then they wanted a second page where it was more general information. Okay, so a lot more space, forced us to be parsimonious. Less is more, but quality in terms of the information that we provide. Here are the graphics. We built upon this based on our experience of doing report back in the Cape Cod and Richmond household exposure studies, and every time we sort of do this, we get better and better and try and improve our designs. So, this is looking at mothers' levels, babies' levels, mothers' levels, babies' levels. The colored circles are the study participant. This is from NHANES data, and if there's a regulatory benchmark, in the case of lead, we stick it there so they can see whether they're quote-unquote high or low in absolute terms. So, and then if we didn't find anything, we just left it blank. So I point these things out to you so that you can see that after people looked at these charts, we also did some tweaks here, not quite as dramatic. But we changed wording, so instead of using benchmark, we used level of health concern, and then we put the words not found here instead of just leaving it blank because people didn't quite know what to make of that. So I think the one thing that this kind of getting input from study participants, and whether it's just a biomonitoring study that is not community engaged, it's more, I would say, kind of an opportunistic exposure study, or a community engaged project like the household exposure study that we did in Richmond and in Cape Cod. Really, the strategy around report back is to think about how we can link results to kinds of actions that people can take, either as individuals in terms of, for lack of a better phrase, shopping your way out of the problem, okay? But also acknowledging the limitations of shopping your way out of the problem, because many times you just can't do that. And also highlighting opportunities for people to take collective action, okay? If those are in fact available, depending on the contaminant. So we've always tried to kind of keep these two things in mind when we design our protocols. In terms of participants' reactions to viewing these materials in the usability testing phase, these are sort of some nice quotes. I got a lot of information here that I didn't know already. The study people already told me things, but there was a lot more here. The reason why people get cancer might be here. I grew up in the 70s and 80s, and they didn't know much about all this. Maybe something back then contributed to my future bad health. The summary pages were very interesting. Women were able to identify their own results in the charts. They seemed to navigate pretty well through them. They could identify whether they were high or low relative to NHANES data or relative to some benchmark if that was available. And then another participant said, at first I'd think, oh my God, there's a blue circle. Then I'd see that for some, none were found. Then I'd see where I was compared to other ladies in the national average. Then I'd see how I'd feel. There's still some challenges in terms of making this information accessible and useful. We did usability testing with a subset of chemicals. We talked a lot with women about the extent to which, we test for so many chemicals. We want to make all the information available, but lots of compounds, it starts to look like alphabet soup, PBDEs, PFCs, PCBs, blah, 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 blah. I mean, I have a PhD, I can barely keep track of all the, congeners of all these different things. So it can be a little mind-numbing, I think, when you have lots and lots of this stuff. So figuring out how to facilitate people's ability to focus on the things that they think are most important to them and maybe skim the rest, I think, is always a challenge. Understanding how their results compare to different kinds of things to make it useful to them. And then the other thing that we found is, for our Spanish speakers, when we were doing usability testing, we really had to work with them to encourage them to give us feedback. They were more reluctant to tell us that this thing was kind of stupid and didn't really work. And so there was a lot more conversation to encourage our Spanish speakers to be honest about the extent to which things were working or not I think the other take-home message is that people need a diversity of ways of taking in their results. They need pictures. Some people like pictures. Some people don't like pictures. Some people just want words. And they want opportunities to drill down to get more detail if they want it, and they want to have options to skim through, as I mentioned earlier. The other thing is, the law in California for reporting back says that people have to get their actual values, so the actual level. So as you can see from the charts that I showed you before, we had the level on the bottom of the chart. But when you talk to participants, they're not necessarily interested in the actual number itself because it's less meaningful. What they want to know is where they compare with other people in the study group, where they compare to a national average. That's how they think. It's always, for them, high and low was relative to something else. The actual value was not, parts per million, micrograms per deciliter, not quite as meaningful to them. And always to have the ability to have a resource person to ask questions. So now we're processing, we did report back to women, and we did interview the women who actually got their real results, not looking at fake data and asking them. And so these were sort of just preliminary reactions, and it really spreads the gamut. So when we asked women, were you surprised with your results? We had people who said, no. I know the world we live in. In other words, basically, I know that we're exposed to a lot of chemicals. I know the life I live, and I expected that you would find something. And for other people, it's much more of a learning experience in terms of really wondering. It's like, why do these chemicals enter my body? I don't work in an obviously, place where I would expect to have these kinds of things. I don't have these things in my home. And so you can kind of see that thinking process and awareness about the ways in which we come in contact with environmental chemicals and the extent to which we don't necessarily have a lot of individual control over our exposures, particularly to certain kinds of consumer products. So we've talked a lot about the ways in which community-engaged science is really great for community partners. Okay, I'm almost done. And I think I want to also talk about the ways in which the co-production of this kind of scientific knowledge and how we deliver it to people and work on how we're going to deliver it together by engaging participants in the design of study protocols promotes what I like to call the 3 R's in terms of making science better. It makes science more rigorous because it helps us improve our research designs, helps with retention and recruitment, helps with all kinds of things. We can talk about that more later. And it's always making sure we're asking the right questions. As you can see by those first materials that I showed you, they were bad, but we thought they were great, okay? And then they just made sure we were, doing better. And then always thinking about how we leverage this knowledge and help people use the knowledge they get about their exposures to promote policy change. And the last thing I want to leave you with is we work in a context as scientists, as community partners, where there are structural inequalities. We always have to keep our eye on that and sort of always be working on it. There is still, we live in a situation of institutional racism. We have a situation where predominantly white scientists are often working in communities of color. So always being aware of that when we're kind of working together and designing protocols, and what the implications of our relationship is in our partnerships and collaboratives is key. Navigating these IRB challenges, and ultimately, how do we decide what's going to happen with the data once it's generated? And I just wanted to leave you with this continuum. Many of us work here, but I wanted to emphasize with this talk today that even at this level here, you can still engage participants and make your science better. And thank you.
Wonderful. Thank you so much, Cecile and Rachel. So we're going to take about 10 minutes for questions. We're running a little bit behind on schedule. I don't know if you'd like to sit up here or stay where you are. Okay, great. And Jacob's got the microphone, so if anyone has a question, just raise your hand.
This is a question for Rachel. I think that was a fabulous study and a great example of how to conduct the research. I'm just curious, were you required, if you identified somebody who had actionable clinical levels, did the grant or the process somehow help this? I mean, these are low-income people.
Yes, actually we did identify a few participants, or actually one participant in particular who had pretty high levels of mercury. And so we were able to work with the San Francisco Department of Public Health to try and track what was going on. And it took a while because we were like, is it the fish? And we were like, you know, went to the market and bought fish where she bought, you know, and it turned out it was hand cream that had been doctored. She had purchased it in Mexico. It was Pond's cream, and you take it to a pharmacist and turn it into a skin lightener, and it has a lot of mercury in it. And so we found it in her bathroom. We used, and she had it on her hands. We test, you know, we could test it. We could see it on her hands because she applied it every day. She didn't want us to test the baby, kind of understandable, but we were able to identify the source, and we had the resources to do so.
I think that's really interesting. I'm wondering what your process was like for choosing which 50 chemicals to look for in this group of women and babies.
The process was, the chemicals that were chosen were based on the California Department of Public Health Lab's capacity to, and what they could analyze, as well as Department of Toxic Substances Control. So there was a lot of interest in flame retardants because we were still in the process of trying to ban and phase out the use of a lot of the PBDEs. And so we were trying to, and at that point there had been relatively little done to assess the levels of exposures in prenatal exposures and also exposures in neonates. So that a lot of it was driven by policy considerations, interest in the lab in developing methods to test emerging contaminants. Those were kind of the primary drivers by the biomonitoring program in terms of what they wanted to analyze. And then of course general interest in the types of things that the Department of Public Health tracks on a regular basis, like a lot of the metals.
Hi, I'm Keely. I'm an anthropologist at the EPA in the Office of Research and Development. And I have a question for Cecile. Great presentation. Really resonated with a lot of things I've encountered. My question is, when you were talking with the social science, did they have any particular strategies that they thought were more or less effective in terms of getting their work included, or their voices heard, or framing the problem, any of that sort of thing?
Yeah. So on the strategy, first of all, they had to join forces. So most of the time, they were really working interdisciplinary among themselves. So if there were an economist and a sociologist, they will sometimes stop the fight that they will have outside of the agency and join forces to talk with our scientists. Second of all, they find some entry points, so some topics where they feel there are more literature from the social science and they can really do things. They try to ally with civil society, to align with the stakeholders' inclusion and those kind of programs. But this can be also bad for them, because they can end up, as I said, at the end of the pipe doing the communication. And I think they also join forces now at European or international level. So they share knowledge among themselves. Because there are so few of them, they have interest to participate in joint studies and those kind of things. But most of the time, they were frustrated. They had tested a lot of strategy, and I had people kind of desperate over the phone sometimes, explaining their frustration and the fact that they tried a lot of strategy. Something I would like to say is the political context was also very important. So for instance, in the UK, under Blair, there were loads of things done to include social scientists. And when I started my work, it was the end of the Blair era. And so all the funding were cut, and there were a reorganization process, and all the strategy that were successful for them before, I could see that from one day to another it could stop.
Great presentation, so thanks. Rachel, do you have any sense that any of the women in the chemicals in our bodies then got involved in any kind of policy work? Did you do kind of a model, or was it always just kind of individual actions and stuff like that?
In our interviews, none of the women indicated that they were that they had been motivated to get into policy work. Although many of the women had sought to kind of make changes in terms of the kinds of products they use, and they had also kind of expressed surprise and frustration that there's not better regulation of environmental chemicals and products. But we didn't, in this particular group, we had not gotten evidence that anyone had decided to become active on the issue in the traditional sense of advocacy or activism.
You just keep thinking it needs to be in the California Biomonitoring Program, some kind of next step that's not necessarily mandated by regulation, but more interaction with the data and the communities of concern that were tested with groups on the ground could then take the data and say, all right, here's some things that could be done and do some kind of outreach with the people that were tested. And we don't know who they all are, but there could be ways to maybe put up flyers and say if anybody wants to talk about this further, about the results, not necessarily the participants, although that would be one of the targets. Try to take that data and maybe make it more useful in terms of getting some changes there, because it seems like one of the intents of Biomonitoring California that communities would be biomonitored, and community means that could be many things, but it means that there are groups of people that are at least talking to each other with concerns, and which is a different model than the CDC and Haynes, which is random populations doing things. And those, they're both very important, but I'm just saying, okay, we've got the data, let's move with it. It's always a thing.
So I'm curious, actually, because Birgit, I don't know if she's in the room. There you are. Okay, I thought I saw you. So I think what you're suggesting, Charles, is great, and I totally agree with you. But when you're working with a state agency, there's certain restrictions around and government agencies that you're operating under that you might not have if you had your own, federally funded project, right? So I'm just kind of curious the extent to which you run into that in some of your experiences with report back.
But in the COFES DEMOC office, it was really very well structured for all the communication before, during the project, after the project.
Yeah.
And we all reported back in a kind of way as you told us. First, it was the research participants. They got a letter with their results, and it was indicated like a kind of traffic lights where they are, where they were situated in their own results in comparison with the others. They had a separate information leaflet, and so it was really well documented. And then also at national levels, it came after that, a few weeks after that, there was a day on environmental health. There were a lot of campaigns. It came in the newspaper. And then a third level, it was at the European level. And then more policy got involved. Also, European Chemical Agency, the Food Authority, these big kind of organizations, they were all closely involved.
To what extent do you think the participants had a level of awareness and might get engaged in these issues once they participated in this kind of program?
Yes, absolutely. It's
yeah.
Great. We have time for one more question.
I appreciated both presentations. I've seen versions of yours, Rachel, of course, but I was really interested in that sort of ramp up visual towards the end of from the low end of participation to high, and thinking about that as temporal, and advice you may have for studies that may have started in the left side and how they might ramp up, or maybe from study to study, what sort of processes or capacity building would be useful to help move up the line more towards the CBPAR.
Well, I think there are some nice examples out there of studies, particularly cohort studies, that have evolved in that way. So my colleague Brenda Eskenazi, who has a 15-year cohort, the Chamaco study, which I'm sure many in this room are familiar with, when they started out, they were not reporting back results. No, I mean, nobody was. It was something that just people didn't think about. And then they decided to kind of put their big toe in the water and try a passive report back system where you would tell people they had results, and if they wanted them, they could call a researcher. And they found that not very many people took them up on that. And then they decided to sort of ramp it up even more and tell people when they came into the clinic, we have your results here, do you want to see them? And then now, you know, they've engaged us, so a lot of the interviews and stuff that I showed you with the California Biomonitoring Program, they have a very engaged process now around report back. They have really gone through a lot of iterations with their IRB to allow them to do it. They're very committed to it. And now they're very committed to evaluating what it means to their participants in terms of their relationship to the study, getting input in terms of the extent to which they have found that information useful and how they can make it more useful. And also evaluating the extent to which participants are really benefiting from a lot of the research translation and outreach activities that they do as part of their bigger Children's Center grant. So I think that is one really nice example of how that's evolved.
Great, thank you so much for a great panel. Oh, yeah.
I just wanted to add a comment because here we hear a lot about the community and the people, but I was wondering if people also interview the lobbyists, the industries, and so on. There are also lots of fieldworks to do on that side. And when we work in France, for instance, for the agencies as social scientists, we really do the fieldwork on every site. So we will have like a mother coming to us to show us that they are burnt harm because they were sitting on the sofa, but we will also have the producer of the sofa who is spraying the sofa with products. So to me, it would have been interesting also today to have people also from the industries, also from lobbying, that try to mobilize science in another way. And I think that opening up could be really interesting in the process.
Yeah.
Great, please join me in thanking our speakers.
So we're going to actually skip the next break in order to make sure that we have lots of time for the things we want. But before we have the environmental justice organizations as collaborators panel start, I want to briefly bring up Margot Golden, who is the chair of the board of the Mass Breast Cancer Coalition. Some of you may have seen her display over there. And MBCC has been a very important group, as you heard, in terms of the founding of Silent Spring years ago. And she'd like to say a few words about the communications partnership that's relevant to our curriculum project. So we'd like to welcome Margot Golden.
Thank you, Phil, very much. So I'm Margot Simon Golden. I'm president of the board of Massachusetts Breast Cancer Coalition. As Julie Brody has mentioned, and Robin, both from, and Herb, all from Silent Spring Institute. In 1991, Massachusetts Breast Cancer Coalition came together. They were concerned with the high incidence rates of breast cancer in Massachusetts, being higher than some other states in the country, with Cape Cod having a very high incidence rates. Their concern turned into rallies and getting together. In 1994, they decided to make a lab of their own and founded Silent Spring Institute. So now we are doing the research. So what do we do with the research? With the research, what Silent Spring, our sister organization, as they do the research, Massachusetts Breast Cancer Coalition does some of the community outreach, community education. So two of the things that we're doing now, presently, along with things we've done in the past, with the collaboration of things like this, conferences. But right now we have two things going on. We have one that's on the table, this Let's Talk Prevention, reducing toxic exposures. So it's a tour that's going around the hospitals, the outreach into the community, wherever that may be, libraries, even health food stores. We're going everywhere that asks us to come, mostly hospitals, but really everywhere across the state. The other thing that we're doing is we have a video. So we have Phil, we have Julie Brody, we have Robin Dodson, we have myself. I'm health affected. I have breast cancer, 14 years, stage 4. That's the powerful story that everyone keeps saying you need to have the powerful story first, and so people can relate to it. And so we have Robin, Julie, and Phil talking on NBCC.org video series about the research that they're doing. And we start from everything from the rallies. We have someone talking about the rallies in the beginning to the studies that are going on now. So people can, and why we're concerned. What's the link between environmental contaminants, breast cancer, and other diseases? So the videos themselves are more of a community outreach.